Okay, well, welcome to this month's Netchatter, and I will pass you over to Dr. Del Rivero to introduce this month's speaker.
That's me, right?
That's you. That's you. That's your name.
Okay, thank you so much. Well, welcome everyone. And it is my pleasure to introduce our speaker for today, Dr. Rachael Guenter. Dr. Guenter did her Bachelor of Science at the North Carolina State University in Raleigh, North Carolina. She then subsequently did her PhD at the University of Alabama, Birmingham, in the Department of Surgeries, and graduated biomedical science program. She also she received as well a lot of amazing training under Dr. Chen's lab. And currently she is under the mentorship of Dr. Bart Rose. Rachael's graduate students has been dedicated to investigating the molecular mechanism of neuroendocrine cancer, as well as developing novel therapy strategies to treat neuroendocrine cancer. Rachael has multiple publications in the field of neuroendocrine cancer biology, including strategies to improve the detection of neuroendocrine tumors. Her recent focus has been about the understanding of the role of Notch signaling, as well as Notch signaling pathway in gastroenteropancreatic and pancreatic neuroendocrine tumors. She has a very particular interest in develop new therapies for patients with unresectable and advanced disease. And as we all know, this is an unmet need. She's also an advocate for patients too. She brings awareness about neuroendocrine cancer. She also aims to provide accurate information for patients as well as the general public. And with that, it is an honor to introduce Dr. Guenter today, and the floor is yours.
Thank you so much, Dr. Del Rivero, for that kind introduction. And I'm very excited and honored to be here to speak to you guys. So today I'm going to be talking about one of my scientific projects in the lab. And it is kind of science heavy, but I'm going to try and make it digestible. But I invite anyone to please interrupt me at any time if you have any questions or something isn't super clear. So the project that I'm going to talk to you guys today about is called Targeting Notch Signaling in Neuroendocrine Tumors. And I started this project back in 2019, and I've been working on it ever since. So for today, we're going to talk about a little bit of background so I can kind of explain what we're doing and maybe why we're doing it. Then we'll get into some data from some experiments, and then we'll end with some conclusions learned so far. So everyone here definitely knows what neuroendocrine cancer is. But I always put this slide into every talk I do because I make a point to, tell everyone in the room what neuroendocrine cancer is so that more people are aware of it. And so we all know it's one of the fastest growing classes of cancer. It can be found everywhere throughout the body, different organs. Some of the neuroendocrine tumors can cause symptoms in patients from the hormone secretion, not all, but some. Neuroendocrine cancer is often misdiagnosed. This statistic actually says it can take 5 to 7 years on average to reach a diagnosis, but that does vary. And then some patients are stage 4 by the time that they finally received an accurate diagnosis. And so another big topic that I want to talk about that's kind of going to be the spotlight of my talk today is this biological signaling pathway called Notch. So this pathway was discovered back in 1917 by researchers who were studying fruit flies. And so they did a mutation in the DNA, and they found that after doing this mutation, the fruit flies got this notched wing appearance where part of the wing was taken out. And so, lo and behold, that's how Notch signaling got its name, was from this defect in the fruit fly's wing. And so since its discovery in 1917, we've learned a lot about Notch, still more to learn, but so far, it's pretty well accepted that this pathway has been evolutionarily conserved. So it's a very old pathway, and it's found in all metazoans, and it is used for cells to communicate to one another, so cell-cell communication. And so when these cells are communicating using Notch, we know that this pathway can regulate cellular differentiation, proliferation of the cells, and specification. So the next thing I want to bring up is the machinery that's involved in Notch signaling. And so to start, Notch has 4 proteins that serve as receptors that are in the cell membrane. So if we pretend this dotted line is the cell membrane, each of these 4 receptors that are conveniently called Notch 1, 2, 3, and 4, that there is an intracellular piece of the protein that hangs out inside of the cell. And then there's an extracellular piece that is outside of the cell. And that extracellular piece is what's going to interact with some of the 5 ligands that can be located on another cell. So similar to the receptors, these ligands are located in the cell membrane, and there's an intracellular piece and a piece outside of the cell. And so these ligands can then interact with the receptors between cells. And so I'll show you kind of what that looks like. So in this diagram, we have 2 cells. The cells are like these tan circles here, and this is the cell membrane in the dark tan. And then the blue is like extracellular space. So one cell is going to have its Notch ligand. And so there's going to be a piece on the outside that can physically bind with the Notch receptor on another cell. And so when this binding between the Notch ligand and the Notch receptor occurs, there is a minute physical force that happens that can actually trigger a cascade of events. So what do those cascade of events look like? So that physical force between that binding is going to cause a conformational change, which is then going to eventually cause these scissors, which are actually, it's a protein, it's an enzyme called gamma secretase, but that's not important right now. But this protein is going to act like a pair of scissors, and it's going to come in and it's going to cut the Notch receptor off of the cell membrane. And then that piece that got cut off is going to travel all the way to the nucleus of the cell. And then in the nucleus is where the DNA is stored. So once the Notch receptor that's cut gets to the nucleus, it's going to bind a couple other proteins, and it's going to ultimately then bind to the DNA, and it's going to regulate whether certain genes are turned on or certain genes are turned off. So this is a rather powerful aspect of Notch signaling because it can have a lot of influence of what's going to happen to the cell by controlling the expression of different genes. So some of the classic genes that are known targets of Notch signaling are called Hess and Hay family genes. And these genes can then go on and affect other genes. For example, one of those is ASCL1, which is a neuronal transcription factor. So this is where we start to see it could come into play with these neuroendocrine tumors, because Notch may be coming to the nucleus here and controlling the expression of genes that regulate neuronal or neuroendocrine features. So two other bits of information I want to throw in is that previous studies have found that there is indeed evidence that Notch, specifically Notch 1, kind of from here on out, I'm going to focus on Notch 1. I also do look at the other notches, but I would have you guys in a 6-hour meeting if we went through all 4 of them. So we're just going to stick with Notch 1 for now. So there is studies out there that have shown that Notch 1 plays an important role in neuroendocrine tumors. And the other key bit of information I want to mention is that we can acknowledge that some therapies, like somatostatin analogs, have demonstrated the ability to prolong progression-free survivals. We still need new therapeutic options. And so with all that information taken together, it has led me to this hypothesis that potentially Notch 1 signaling contributes to the growth and survival of neuroendocrine tumor cells. And so for this talk specifically, I'm just going to focus on the pancreatic neuroendocrine tumors. But I do think that Notch signaling plays a role in more than just the pancreatic neuroendocrine tumors. Okay, so now we're going to jump into some of the experiments and some of the data that I found so far in studying my hypothesis. So one of the first things that we looked at to see if, Notch was in neuroendocrine tumors and what in the world could it possibly be doing was to look at its expression in some neuroendocrine tumor samples from patients. And so to do that, we can do these different types of stains or dyes on the tissue. And so here I did two different stains. And so one of them is the color blue, and this is a chemical called hematoxylin, and it binds DNA. So that helps us see where the cells are, and we can see these dark blue circles, and those are the nucleus of the cell where the DNA is stored. And then the other stain is for specifically the Notch1 protein, and that color is brown. So whenever we see brown, we know that the Notch1 protein is present. So what you'll see here is we took islet cells. So these are normal islet cells in the pancreas, so the endocrine cells in the pancreas. And I stained them both brown and blue, but there is no brown color because there's no Notch1 there. So in these normal islets, Notch1, not there, not expressed. However, when I put both stains on a metastatic pancreatic neuroendocrine tumor, now we can see both colors. You can still kind of see the blue nuclei scattered throughout. These are all the different cells clustered together. And now we can see the color brown because this pancreatic neuroendocrine tumor has Notch1. What is Notch1 doing there? Well, we'll see. But it's there, and it's not normally there. And so then to take it a step further, we used this thing that we built this at UAB, and this is called a tissue microarray. So each of these little circles that you see is actually a tumor from a patient. So almost similar to like a biopsy. So I believe this tissue microarray has 38 patient tumor samples. And so we take like a little core punch out of the tumor, and we put all of these circles on one microscope slide. So when you're looking at this with like your human eye, it's very small because we've put all of these tumor samples on one microscope slide. And then I stained this microscope slide with all 38 tumor samples for both the brown and blue stains to see if Notch1 was present in any of the samples or how many of the 38 tumors had Notch1. And so what you can see is that there are some circles or some tumors that are totally blue and they don't have Notch1. But then again, there are some circles, meaning some of these patient tumors do have Notch1. And so I kind of zoomed in down here at the bottom to show you an example of some of the pancreatic neuroendocrine tumors that were brown, meaning they had Notch1. So from this, I can conclude that some tumors, some pancreatic neuroendocrine tumors, some patients do have Notch1 and some do not. Okay, so then the next step was to figure out what Notch1 was doing. And so I always share this example of how I got to this process. So back in college, when I did engineering, there was an example that one of my professors was telling us about, and I don't even remember what the context was. But what he was saying was trying to figure out how something works, but he was talking about it in terms of a chemical manufacturing facility. And he was like, if you get rid of something, you will quickly find out what it did. So his example was a car. So he was like, if you were an alien and you came to Earth, you got in a car and you started going and you took off the steering wheel, you'd figure out what it did. And then if you took out the brakes of the car, you would very quickly figure out what the brakes were for. And so that was kind of the logic that I was thinking of in terms of trying to understand what Notch1 was doing in these tumors. I was like, well, if I just completely get rid of Notch1 and the cell can't use it, even if it wants to, then I'll have a better chance of figuring out what this protein is doing. And so that's what I did. And so in order to completely delete Notch1 to where the cells couldn't even use it if they wanted to, I used a technology called CRISPR-Cas9. And so this is a genetic engineering technology where you can kind of manipulate DNA. And so you can have a target sequence here in orange-ish, yellowish. And so in my case, my target sequence was Notch1. And then you can design these guide RNAs, so this string of nucleotides that will specifically bind your target. So I did that for Notch1. And then you can add this protein called Cas9. This is another enzyme. And then Cas9 is going to bind to the guide RNA. And once Cas9 gets there to the DNA, it's going to make a cut. It's going to do a double-stranded cut on the DNA. It's going to cause a breakage. And so when Cas9 cuts the DNA, that's going to introduce a lot of mutations. It can take out DNA. It can be almost detrimental to that gene, to where that gene is going to no longer be functional. And so a protein's not going to happen, or protein's not going to be able to be made. And so I'm going to show you kind of, my process of doing this in the neuroendocrine tumor cells. And so this was the workflow of how I got rid of Notch1. So I first characterized my neuroendocrine tumor cells. These are from the pancreas. This is a cell line called BON. BON is just the name of the cells, these pancreatic neuroendocrine tumor cells that we grow in petri dishes in the lab. And so I took the BON cells, I designed some guide RNAs against Notch1. And then I added the guide RNAs and the Cas9 protein to the cells. And then I looked at all the different cells. So different cells had different mutations based on kind of chance. How many Cas9s got to the cell, did the guide RNAs work? And so I screened all the different cells and the different types of mutations that occurred. And then I found one that was a pretty hefty mutation to where I think Notch1 was deleted. And so then I took that group of cells and I put one cell in a single petri dish, and I let that one cell grow up into a bunch of cells. So this is called single cell cloning. So right here, this started as one cell in the middle, and now it's dividing into a whole island of cells. And then I did DNA sequencing to understand the mutation and what happened. And so what I found, so this is the DNA level, was that there is a piece of the Notch1 gene missing. So WT here means wild type. So this is like normal, unedited, just wild type. And then KO is knockout. So wild type Notch1 gene is just what I sequenced here, just below 500. And then the knockout, it's much lower because there's a piece missing, which I'll show you a diagram of what happened in the next slide. But then to further make sure that I did delete Notch1, because it looks like, yes, the DNA is altered. There's a piece of the Notch1 gene missing. But what I'm really concerned about is the protein that comes from the Notch1 gene. And I wanted to make sure that the protein was gone too. So to do that, I did this experiment to look at the protein level. And so I took, again, the wild type BON cells, not edited. I looked at them vehicle, meaning control, no treatment. And then to make sure Notch1 was not there and could not be there, I treated with a drug called VPA that is known to turn on Notch1. And I did 2 different doses of VPA. So in the wild type normal conditions, what we see, there is a low level of Notch1 to begin with these cells, but they can turn it on when they want to turn it on, essentially. But then I added the VPA to force the cells to turn on Notch1. And there, because of these black lines, we can see that Notch1 got turned on. But then I tested this in my knockout cells. And what I found was, again, there's no Notch1 to begin with. And even when I add the drug to the cells to force them to turn on Notch1, they can't. There is no Notch1. And so all of this taken together, I figured out that I had successfully deleted Notch1. And this is a schematic of what I believe has occurred based on my sequencing and analysis. So normally the Notch1 gene is found on chromosome number 9. And you have 2 different alleles here. And then I did the CRISPR-Cas9. And then that led to, on one allele, a 65 base pair deletion. And then on the other allele, 468 base pair deletion. So both alleles were affected. The 65 base pair deletion is actually a frame shift mutation. Since DNA is read in multiples of 3, 65 is not divisible by 3. It's a frame shift mutation. And so these two edits from the CRISPR was enough to cause what we call a compound heterozygous knockout of Notch1. So the next thing is, I know what you guys have all been waiting for. What happened? How did the pancreatic neuroendocrine tumor cells change now that Notch1 was deleted? Okay, so I want to talk about one of the first things I noticed. And I remember talking about this to a bunch of scientists, and they were like, at first they really didn't take me seriously. They were like, this doesn't mean anything. But actually, I think it does. I'm very receptive to how the cells grow. I'm very aware of what they're doing. And so I noticed this phenomenon that I'll share with you. And so I'm growing these pancreatic neuroendocrine tumor cells in a Petri dish. And what you're looking at right now is our wild type, our regular pancreatic neuroendocrine tumor cells. So I put them in a Petri dish, and then I started taking pictures around day 6. And so these are the cells here, this textured looking stuff. And then there's background, that's just empty space. It's quite literally plastic, because the Petri dishes are made of plastic. And so there's some empty space. And then so by day 9, the cells are growing. And so then there is no plastic, there's no empty space at this point. The cells have completely covered the bottom of the Petri dish. And what these regular pancreatic neuroendocrine tumor cells will do is, once they've covered the whole bottom of the Petri dish, is that they'll actually start growing in 3. They'll start stacking on top of each other in layers. In fact, they'll even form something that we joke about in the lab. We call it a spherical creature. They'll start building these 3D masses of cells. So nothing, even if they don't have any more room to grow, they'll just start growing on top of each other. But what I noticed was that when I deleted Notch1, this stopped happening. So these are these are the pancreatic neuroendocrine tumor cells without Notch1. And so you can see them, the textured, and then there's the background. And then by day 9, they've covered the bottom of the Petri dish. And then as time goes on, they stay there. They've covered the bottom of the Petri dish, but they're not going to stack on top of each other. They're not going to keep growing. They're going to stop growing. And so I wanted to be pretty scientific about this, instead of, telling other people, oh yeah, they stopped stacking on top of each other. So I wanted to kind of quantify that. And so I did an experiment that is in 2 different parts here, part A and part B. So part A, I measured viable cell density, or basically the number of cells that are alive over time, in the regular wild type cells and the cells that didn't have Notch1. And so what you'll see is that at a certain point in time, the cells will hit what we call 100% confluence or confluency. It's 100% confluence is when the cells cover the entire bottom of the Petri dish. And so both of the cell types reach that. And the wild type cells, once they reach that, they keep growing. There are more cells that are alive because they're stacking on top of each other. But when the cells without Notch1 cover the entire bottom of the Petri dish, they stop growing. They just halt. And so interestingly, I also looked at the expression of Notch1 in these wild type cells. I can't look at it in the knockouts because it's not there. But in the wild type cells, we see some Notch1 in log phase. So log phase is when they're actively dividing. And then I call it kind of overconfluent, when they're stacking on top of each other. And when the cells get in that condition, they have a significant increase in the expression of Notch1. So it seems like Notch1 is doing something to help these cells continue growing and stack on top of each other and be more aggressive. Okay, so then I looked at something else. I looked at the ability of the cells to migrate. And so what do I mean when I say migrate? These are in a Petri dish. So I'll show you an example of this experiment that we do. So this is a plastic chamber called a transwell assay or a Boyden chamber in gray. And then there's this membrane scenario in blue. And then what we can do is we can add cancer cells on the top piece here. And so the cancer cells are here in yellow. And we add them in media, or it's essentially food for the cancer cells. So we put them in there. And then we also put media or the food in the bottom half, in the orange. And so what we'll do is we'll actually kind of starve the cells that are in the top, and then we'll add extra nutrients to the media in the bottom half so that the cells want to crawl towards those nutrients. And the blue membrane here is porous. So there's little pores in it. And so the cells can crawl through that membrane and get to the nutrient-rich media on the bottom. And so this is a way to test the ability of the cell to migrate or to essentially crawl. And this can be important because it can be a measure of metastasis. I mean, this obviously in a body, we don't have a membrane that cells are crawling through quite literally, but this can almost serve as a proxy to test the cell's ability to move. And so then you wait a certain period of time, and over time, the cells will crawl through that little membrane. So this is the experiment I did in the wild type pancreatic neuroendocrine tumor cells, and the cells without Notch1. And so I did this experiment, I gave them time, and when they got to the other side of the membrane, or if they got to the other side of the membrane, I then stained them with a chemical that turns them blue. And so what you'll see is here's a picture of the bottom half of that membrane, and we see that the regular wild type pancreatic net cells, several of them crawled through, and you can see the cells are in blue. And then the cells without Notch1, way less cells were able to crawl through that membrane. And then I did a graph here to quantify that. And so then this tells us that Notch1 must be helping these cells in their ability to migrate. So the cell's ability to survive and grow and proliferate and migrate, these are all aspects that we don't want cancer cells to have. That's what makes them aggressive. And so my thought is, okay, if Notch is playing an important role in this, then maybe we need to be toning it down. Maybe we need less Notch if Notch is helping these cells be more aggressive. And so I wanted to find a way to block Notch signaling in these tumor cells that wasn't CRISPR genetic engineering, because they can't be doing that. And so there's actually a class of drugs that can be used to block Notch signaling. And so I'll briefly show you how that works. And so we remember here, this is the mechanism of Notch signaling. So the two cells will interact with their ligand and receptor. This enzyme that acts like a pair of scissors comes in, cuts the receptor that sends it to the nucleus to where it can control gene expression. So this class of drugs called gamma secretase inhibitors will actually block the activity of this enzyme that acts like a pair of scissors. And so the receptor never gets cut. And so it never comes off the membrane, and then it never goes to the nucleus. And so this is the most common class of drug that is used to block Notch signaling. And so of course I wanted to test this in the pancreatic net cells. And so these graphs are a little heavy in terms of data, so I'll walk you through it. And so for this experiment, testing these gamma secretase inhibitors in the pancreatic net cells, I used two different cell lines of pancreatic net cells. So one is called BON, and then there's another cell line that we can grow in Petri dishes called QGP. And so I wanted to test them in both. And these are actually basically the only two cell lines available to study pancreatic nets. And so there's a big push to develop more cell lines than just these two to study. So it's two different cell lines. I treated them with actually 3 different types of gamma secretase inhibitors. And then I also treated them with lanreotide, which is a somatostatin analog and often first-line therapy for patients. So I kind of used that as a comparison. And so the line with the black squares is lanreotide. And then I used a drug called Neuroendocrine, which was developed by a company called SpringWorks that I believe was eventually bought by Pfizer. And then another gamma secretase inhibitor called DAPT. This one is kind of the most popular gamma secretase inhibitors for those who study Notch signaling. It's been around for a really long time. And then the third gamma secretase inhibitor is called MK0752, and this is a compound developed by Merck. And so what I found was I really, coming into this experiment, I would expect all the gamma secretase inhibitors to act the same way. But interestingly, what I found was that this one called Neuroendocrine, in the red dots, was the most effective of killing the cells. So the y-axis here is the percent viability, or the number of cells that are still alive after different amounts of drug. And so as we add more and more drug, Neuroendocrine kills more and more cells. And so for moving forward, I selected that gamma secretase inhibitor to continue testing it, because it appears that it has unique ability to affect these pancreatic neuroendocrine tumor cells. And so then I did another experiment in both cell lines where I combined lanreotide, the somatostatin analog, which is normally first-line therapy, or one of lanreotide, octreotide, the somatostatin analog, with Neuroendocrine, that gamma secretase inhibitor that I saw was working quite well in terms of killing cells. And so in both cell lines, the black line here is just lanreotide by itself. So add more lanreotide, measure the number of cells, or the percent of cells that are alive. Lanreotide doesn't do a whole lot. Of course, over time, and in a in-person, a human, these are cells in a petri dish, we know that lanreotide can improve the progression-free survival of patients. But in terms of a petri dish, adding this amount of drug just once, we didn't see much of an effect on cell killing from lanreotide. But if I combine lanreotide with the gamma secretase inhibitor Neuroendocrine to block Notch signaling, now we see a drastic decrease in the number of cells that are alive. And so I think this is the last piece of data that I'm going to show, but I really wanted to show this because I think it's really interesting. And spoiler alert, at this point in time, I can't explain to you what exactly is happening here on the cellular level. I am working on that, but I do think it's interesting. So, so the last experiment, I was testing the somatostatin analog lanreotide in combination with the gamma secretase inhibitors to block Notch signaling. And then I also wanted to try a drug called everolimus that can be used as second or third line therapy for patients with neuroendocrine tumors. Excuse me. And everolimus is a drug that blocks another biological pathway called mTOR. And so if I were to combine everolimus in a gamma secretase inhibitor, I would be blocking both the mTOR pathway and the Notch pathway. And so, but before I got to that, I wanted to test it in the regular wild type pancreatic net cells compared to the pancreatic net cells that I deleted Notch1 in. And so I did this experiment where I added increasing amounts of drug, and then I looked at the percent of cells that are alive. And so we can measure kind of a cell's ability to kill cells, drug's ability to kill cells by using this metric called IC50. And so IC50 is the concentration of the drug that it takes to kill 50% of the cells, so to kill half of the cells that you started with. So with everolimus in our wild type pancreatic net cells, it took 43.7 micromolar of the drug to kill half of the cells that I started with. However, when I did the same experiment in the cells that didn't have Notch1, I found out that it only took 3 micromolar to kill half of the cells. So this means that the everolimus is 14 times stronger in terms of killing cells when they don't have Notch1. So this tells me that Notch1 might be doing something to protect the cells from dying when being treated with everolimus. And so then I did end up combining everolimus with Neuroendocrine, the gamma secretase inhibitor, to see if we saw a similar effect. And so what this experiment was the same type of scenario, increasing amount of drug and measuring the percent of cells that are still alive. And so again, everolimus by itself, it took 39 micromolar, which is similar to the 43. There's of course always some natural variability in every experiment that you do. And so in this specific experiment, 39 micromolar everolimus killed half of the cells that were in the dish to start with. But then when we combine everolimus with nirogacestat, the gamma secretase inhibitor, now it only took 12 micromolar to kill half of the cells there. So all of this kind of taken together, I think that there's something going on here that Notch is doing to protect the cells from death, or doing something so that the cells can evade dying when being treated with everolimus. And so all of this taken together, I've come up with 3 conclusions. I've come up with a lot of conclusions, but 3 that I want to share with you. So the first one is that Notch1 may be helping pancreatic neuroendocrine tumor cells grow and survive, and then downregulating Notch1 with gamma secretase inhibitors in the pancreatic net cells may help reduce cell growth and survival. And finally, I think that targeting Notch1 in pancreatic neuroendocrine tumors might be a future therapeutic option, but of course, more testing is needed. And so with that, I have to give a huge shout out to the NET Cancer Foundation. I started working with this organization back in 2020 with Simon. And so I want to say a big thank you to all of our ambassadors that keep us going and help the organization, and all of the patients that are also involved. This organization would not be what it is without the patients, the physicians, and the researchers. Of course, I have to give a shout out to Selena and Simon. And then finally, I have to put a plug in for our upcoming NET Cancer Foundation Research Summit. So this will be Friday, May 19th. And so we'll have, I think, 5 or 6 speakers. So this will be about a 2-hour event, 6 or 5, 2-hour event on Zoom, where we bring together different doctors and researchers from throughout the world. And they'll give about 20-minute talks about what they're doing in the field of neuroendocrine cancer. And last but not least, I also have to say a big thank you to my lab and all of my collaborators that helped me in getting this research done. Of course, my mentor, Dr. Bart Rose, he's a surgeon here at UAB. And then all the other members of the Department of Surgery. Brendan, I apologize. It still says MS. Brendan Herring actually just got his PhD yesterday. So I have to apologize that it says MS on here. That should be PhD. And then the Department of Radiology and the Department of Cell Developmental and Integrative Biology as well. We have a lot of fun outside of the lab. And of course, we always have fun inside of the lab studying neuroendocrine cancer. So with that, does anyone have any questions, comments, concerns, ideas, future directions?
Can I ask a question, Rachel?
Yes, yes.
That was an awesome presentation. And for someone who's not in the lab, it was very clear and I was able to understand everything.
Okay, good. That's the difficult one to explain, so.
I do have a question because I remember on your first slides, you mentioned the relationship with Notch and other proteins like DLL3. And I know that there is ongoing studies with DLL3 for small cell lung cancers and other cancers as well. And I just wanted to understand, what in terms of, it seems like it's also the same mechanism as the gamma secretase inhibitors, where they're trying to shut down Notch so that way other therapies can be more effective. Is that the same rationale with the DLL3 targeted therapies, or what are your thoughts about that?
So it's a little bit different. So in some cancers, especially in the small cell lung cancer, delta-like ligand 3 can be upregulated on the cell membrane. And there's some reports actually that delta-like ligand 3 inhibits Notch1. And so for some reason, some of these tumors are upregulating the expression of delta-like ligand 3. And so they're using that as kind of a targeted therapy with the idea that normal cells, non-cancerous cells, are not going to have a high level of delta-like ligand 3, but some of the tumors are upregulating delta-like ligand 3. So now it's becoming a novel target on these tumor cells. And so they're using it as a way to selectively bind the tumor cells. In terms of the mechanism of once you block delta-like ligand 3, I think that's still being worked out. There are studies that say that delta-like ligand 3 will block the activity of Notch1. So I could foresee a potential scenario where if you do bind delta-like ligand 3 and it can no longer block Notch1, then maybe you get more Notch1 activity. I can't confirm that. That would just be my hypothesis. But as of right now, from my understanding, targeting delta-like ligand 3 is as like a selective tumor marker and less so mechanism in terms of modulating the entirety of the Notch signaling pathway. But we're going to start looking at that. One of my next experiments, I'm going to order some neutralizing antibodies against the different Notch ligands to see how that affects the mechanism of the pathway.
Thank you.
Yes.
Okay, so can you hear me?
I can. Hello.
Okay. Hey, that was wow, a lot of great information. I'm like, my daughter is actually attending UAB. Started off in immunology, but now her path has changed with nurse anesthetist. But anyway, I'm like, gosh, you would find this so interesting.
Well, awesome. Tell her to come by the lab anytime.
Oh, I will. Thanks. But I have just a thought process here. I'm always going, okay, well, if plan A is not working, let's look at plan B, and then let's just talk about plan C. So that's just always the way I'm approaching things. But anyway, so I mean, you're on it with everything that you've shared today. But from that point on, everything that you're doing in the lab, how quick do you take something like this and start practicing on lab rats to see, okay, these are the side effects we're seeing. This because I'm coming from a patient standpoint of view.
Yes.
I'm coming from somebody, oh my gosh, I've had 18 surgeries for not just not just for neuroendocrine tumors, but this is my third time with this. And yeah, I'm just kind of curious, because breast cancer twice, you're like, did you just say chemo? No, that's a bad word.
Yeah.
So I'm just curious, how quick is it? Like, how quick?
Yeah, no, that's a very good question.
And put this into the lab and start, trials or start, I don't know. I'm just curious.
Yeah. So what I can tell you is the rate can kind of depend on the compound that you're using. And so for Neuroendocrine, for example, the drug I was talking about, that I don't want to say it's FDA approved for, I think, like emergency. Has some type of FDA approval for patients with desmoid tumors. So that means that the company and the scientists have already studied the drug extensively for side effects and what it can do and what it cannot do.
Okay.
So as soon as I got these results, I've actually already put it into mice and studied it. I need to continue doing more experiments, but that one, the information was already available, actually because it has been studied in desmoid tumor, which is a different type of cancer. So I was able to know how much to give the mice, how long give the mice. That drug can cause GI toxicity, diarrhea, but that's already known. And they've worked out in previous studies, if you give mice the drug for 7 days, and then you take a break for 7 days, and then you give them another 7 days, and then another break. So you kind of alternate, that can help and reduce any GI toxicity or diarrhea. And so that was already known. So it was very rapid. for me to take it from the Petri dish to do it into mice.
Mhm.
And in fact, we actually submitted it for a clinical trial 2 years in a row, but we did not get funded, unfortunately. So we've gone back to the drawing board to change the plan. And so we did want to see to move that to patients.
Mhm.
And so
Well-
when you're using a drug that's already been studied, it's a lot easier to move quickly.
Oh, yeah. And so when you saw that you did a week, off and on, did you try it consistent? I mean, I know what you just said about the side effects, but did you compare that? I'm just curious, was one like if you did it nonstop versus on off, alternating the weeks, did you see that anyone got healed quicker?
So I didn't do that experiment. Someone else did it in terms of like the mice didn't have cancer, but they did like repeat like nonstop versus like alternating. And the mice at nonstop did have a good bit of diarrhea, and so they lost a lot of weight because
I
of that.
bet.
And so then my thought was like, okay, I don't want to lose a mouse in my study because of diarrhea and losing weight. So I just went with like the alternating one.
I got it.
Be something. These mice then have cancer, so maybe the drug, would be concentrated to the tumor. I think that's definitely something I could
Oh,
try.
absolutely. That's a lot of great information. I appreciate it. I was just
Yeah.
curious how soon you took your
No, I is try and do it as quick as possible. We, when we work with compounds that haven't been studied before, like there was a couple years ago, our lab got a batch of compounds from sponges in Antarctica. They isolated these chemicals from sponges in Antarctica and shipped it to the lab. Jason, am I saying that right? My lab mate's here. And so these were these novel compounds that nobody had studied before. So with guys, that's going to take a really long time. So we have to make sure that they're safe. How much do we use? Do we take it as a pill? Do we take it IV? Do you take it, as a load? You got to figure out all these different things.
Oh, absolutely.
But I have a special interest in, using what's out there to the best of our ability. Of course, we need to find new compounds. We need to keep moving forward, but we also need to optimize what's out there so that we're using them, effectively.
Gotcha. Thank you.
Yes, of course.
Yeah. I do have a question, because basically on the experiments that you have done, it seems that, there is neuroendocrine tumors that expresses Notch. So, and it seems that even if you treat it with a certain approved agent, like you mentioned about the everolimus, it seems that they're unable to kill the cells because Notch is protecting the survival of the cells. So basically what you did is to try to inhibit Notch by giving gamma secretase, and by doing that in combination with the everolimus, then you kind of increase the killing of the cells by doing that. Is that correct?
Yes.
So, and I think that's one of the, kind of like drug development, and this is a question for Mrs. Ingram, because I'm kind of like more on the clinical aspects of drug development, not in the lab. So one of the things is, and it's a valid question, is understand the toxicity of not only of each of the drug, but also in combination. So because combination may have a different side effect profile that if you give it by itself. So that's the reason why in drug development, you have approved agents for gamma secretase inhibitors. You may have everolimus, what is the toxicity. But that's the reason why if you were to do a study, you still need to do a phase 1, because you need to determine what is a safe dose to have the efficacy or some signal that we want before you can do a phase 2 study, where phase 2, you kind of like determine what is more the efficacy of the drug. So still, I think that's a valid question, but yes, I mean, it's kind of like we know the side effect of each one of them, but combination is just, and even though the animals, I mean, I don't do a lot of work, even then animals may tell you something of what maybe the toxicity in humans may be a little different. So that's the reason why we do these clinical studies as well, to understand not only the efficacy, but as well as understand the side effects and the long-term side effects as well.
Yeah, absolutely.
Yeah, but also, I mean, I found it very fascinating because the everolimus and also lanreotide for neuroendocrine tumors, we know stabilize the tumor, but we don't necessarily see shrinkage of the tumor. And I was very surprised to see that when you give the gamma secretase with everolimus, it seems like you do see some killing
It's
of the
maybe
cell.
no longer cytostatic. We start to see.
Exactly, which that's something that I found very interesting. And lanreotide, I mean, is very tolerated. I mean, everolimus has some side effects, but the lanreotide is very well tolerated. So to do that, I think to start, if you want to take it to the clinic, to start something that may have minimal toxicity, but it still has, like you say, some cytotoxic effect, I think that would be very important to do.
Yep, that's what exactly, our thought. And then for the trial that we had wanted to do was combining the nirogacet with lanreotide. And then we had tested that in mice, and we did not see any significant side effects. I measured the mice, their weight every day, and they didn't lose a bunch of weight, and they didn't have bad diarrhea. And we looked at a bunch of other, aspects as well. And we thought about starting at least with lanreotide because, yeah, exactly. Lanreotide doesn't have as many side effects per se than everolimus, and just kind of testing that combination for safety and using a regimen of the nirogacet that would cause minimal side effects.
Mm-hmm. And also, are you planning to do it with like PRT with 177 lutetium dotatate?
Yes. I would love to try that.
Yeah, I think that will be something very good to see, because as we know of all of the treatment options that we have there, Lutathera is one of the options, so we know it can work, but similarly to other agents, it stabilizes as well. Most of the time, stabilize the tumor, we don't see much shrinkage. But if you can do that and with a gamma secretase inhibitors, maybe that will be an option.
Yes, I definitely agree. And then I also I do want to look at other avenues of blocking Notch signaling than just gamma secretase inhibitors. And so I'm going to start looking at, neutralizing antibodies. So adding an antibody that will block the receptor on the outside of the cell and block activation of the pathway through that way. And of course, I'm always reading the literature to see if someone comes out with something new as another way of blocking Notch signaling. Potentially something, since the downside, of the gamma secretase inhibitors is that it's an inhibitor of gamma secretase. It's blocking the enzyme that is causing Notch to become active. It's not technically blocking Notch. And so curious, in the future, if we can find a drug or a way of specifically blocking Notch itself rather than the enzyme that is responsible for active Notch, if it's a similar effect.
Mm-hmm.
So
y'all
I have a
done?
question. Sorry.
Oh, I'm sorry.
I have a question. So when you're talking about it's blocking the Notch signal, so you don't necessarily have to know if someone has the gene for Notch1. Is that correct? Because you would be able to use that?
So I think it would, from a scientist standpoint. More information is always the better. It would be very interesting to know if the patient had the Notch1 protein. But even I think you could still probably use the compound to block Notch signaling, because everyone is going to have Notch1. It's a matter of in our case, the tumor cells turn it on, make it active. The gene is likely going to be there, but I think it's when the cell starts putting it to work. And it can, when the cells, especially the tumor cells, put Notch to work, it's using it to grow and to survive. And so then that's when we want to use the drug to block that activity.
Okay, that makes sense.
So yeah, if there was like an easy way to detect it, that would be great. And in the future, I think they're.
Have y'all done have y'all done any immunotherapy of any sort?
So in our lab, we haven't specifically done any yet. I do think that is such an interesting aspect of neuroendocrine cancer, and a lot of the trials haven't had great results with the immunotherapy yet. And so me as a scientist, I'm like, why? why didn't it work in the
Yeah,
neuro?
I'm like, what are you waiting
Why?
for? That's
Yeah.
like when I was talking to Dr. Reddy, I'm like, what do you mean you haven't taken that from the lab rat to the humans yet? But
Yeah.
I'm like, why haven't you? I'm like, what are y'all waiting for?
So let me tell you, as a scientist, one of the biggest issues right now is that. So what we have to do with the lab rats is those cells that you saw that was growing in a petri dish, those are human neuroendocrine pancreatic net cells. So in order to put them in the mouse, we have to delete the immune system of the mouse. So that, because if we inject human into mouse, then the mouse immune system would just, stop it right there. We have to delete the immune system in the mouse and then put the human cells in the mouse. And that's all fine and dandy-ish until we want to study immunotherapy in the immune system. So we don't have a mouse model. We don't have mice that develop the neuroendocrine tumors with an immune system intact. Well, there is one that we worked on, our lab did this collaboration, but it takes 2 years for the tumors to develop in the mice. And so it's really hard to make progress when we're waiting 2 years for the mouse, 2 years to even see if the tumor develops.
Because you can't just take the tumor and put it in the mouse.
Yeah. We need somehow I tried to, the tumors that took 2 years to grow, I took out some of the tumor cells and put them in a petri dish and tried to grow them so that one day I could put them back in the mice. But they stayed alive for 9 months in that petri dish, but they did not move. They did not divide. They just sat there.
Interesting.
And so that's been a huge hurdle for us making advancements with immunotherapy and neuroendocrine cancer. On the science side is we don't yet have a great way to study the immune system interacting with the neuroendocrine cancer.
Yeah.
We're working on it. We are certainly working on it.
Have you visited other labs or I don't know if that's the right question. I was just curious, because you read in the US and various places that do have some of that.
Yes, and that's an excellent idea, and that's exactly my train of thought too. I am always going to other labs. I'm like, how do you do this? So tell me how you do this. What is your recipe? There's a lab just down from us that studies colorectal cancer, and they grow these things called organoids, where they'll grow the tumors like these giant sphere-like things, almost like tumors, but outside of the body. Yeah. And so I really want to try to see if I can do that with the neuroendocrine tumor cells. So that would be another interesting aspect.
Mm-hmm. I'm sure you'll go far. You sound like you have it. I'm impressed.
We are trying. So yeah, a big push in the field is to develop more models, essentially more mice with neuroendocrine cancer, especially mice with immune systems. That way we can study these things.
Yeah. Thank you.
Yes, of course. These are great questions.
How about the DLK1? What is the relationship between Notch and DLK1?
Delta-like ligand 1. So very interesting. In James Howe's database, where they sequenced primary versus metastatic tumors, like ligand 1 was overexpressed in the metastatic region. Notch 1 specifically is known to interact with delta-like ligand 3. It interacts with delta-like ligand 4 in terms of angiogenesis and vascular development primarily. And it can interact with the jagged ligands as well. I have started looking into Notch and delta-like ligand 1. There are some studies out there, they do interact. What exactly is it doing? I think it depends. And that's one of the most complicated but interesting things about Notch signaling is you've got 4 receptors and you've got 5 ligands. And the interaction, they interact differently with different ones. And so some of the receptors have affinities for certain ligands, but that's not to say that those ligands can't necessarily bind those receptors under. So I think biology and life uses this complexity of Notch signaling to fine tune what it needs. And so it will selectively make those interactions happen based on what that cell needs. And so I think that's fascinating. And so we can continue to study what Notch1 and delta-like ligand 1 are doing, and it may be different under certain scenarios. And so if we can find, what they're doing, and if that interaction is good or bad, one thing that I want to do soon is I'm not going to CRISPR delete all of the ligands, but I think I can use silencing RNA to temporarily shut down each of the different ligands. And so then I can better see what's happening. So I could specifically, use siRNA to shut down delta-like ligand 1 and see what happens to the cells once I do that. And then I recently read another paper with Notch1 and delta-like ligand 1. And so I won't talk too much more, but I've been recently studying metabolism of the pancreatic neuroendocrine tumor cells. Seems like Notch signaling is playing an important role in the cell's metabolism, and that may be reflected in what we see with everolimus, since the mTOR pathway can affect glycolysis. And so I've started going down that road, and there was a paper that talked about Notch1 and delta-like ligand 1 interacting and having a metabolic output. So.
Excellent.
Yeah, I can go down all these rabbit holes with Notch.
I think we are overrunning. I think we are going to have to bring it to a close, but that was really, really interesting. And I mean, I know nothing about the scientific side, and I could follow that really clearly. So thank you, Rachel.
Yay, thank you.
You know how awful I am at following things. But thank you so much. And thank you everyone for coming. So Rachel, Heidi and Kate, don't forget we now have an ambassador meeting very quickly. So thank you very much. And if hopefully all of you, we will see you all at the conference on the 19th of May. It's starting an hour earlier. So that's 5 p.m. in the UK. So whatever that translates to where you are. So thank you very much for coming. I don't know. I don't want to say a time because I always get it wrong.
Yes, thank you all. I will talk to you all soon.
Thank you very much, everyone. Thank you. Bye-bye.